All right, so hello and welcome to the Monday seminar series at the John Mayer USDA Human Nutrition Research Center on Aging. We're excited to have you all join our community for the talk today. And if you're joining for the first time, welcome and thank you for being here. The format for today will be a speaker presentation followed by a moderated question and answer session. And you're welcome to submit questions anytime during the talk in the Q&A box. The topic for today's presentation is related to understanding gut microbiota, which is important in nutrition and healthy aging. Research efforts in this area have accelerated in recent years, especially with the human microbiome proposed as a mediator in numerous aspects of human health and nutrition. So understanding factors that change gut microbiota are extremely important. Our speaker today is Dr. Jenni Firrman. She's a molecular biologist and microbiologist at the USDA Eastern Regional Research Center, which is located in Whitmore, Pennsylvania, and it's part of the ARS Northeast area. She's in the Dietary and Functional Foods Research Unit there. Dr. Firrman does research on in vitro methods to study gut microbiota, and I'm very excited for the talk today. I'm now pleased to introduce our speaker, Dr. Jenni Firrman.
Okay, good morning. It is a pleasure to be here today to talk to you about some of our work where we study the gut microbiota using in vitro methods. As you just heard, I am with the United States Department of Agriculture and part of the Agricultural Research Service. I am located in Eastern Regional Research Center, and this is a picture of our building in Whitmore, Pennsylvania, and I'm part of the Dairy and Functional Foods Research Unit. And I work on a project where we look at studying the effect of diet on the gut microbiota using in vitro systems. And I'm part of a team led by our research leader, Dr. Tomasula, and our lead scientist, Dr. Liu. And then I also work with 3 other scientists and support scientists in our group where we're studying diet, the gut microbiome, and trying to get an idea on how this can affect human health. So I always like to begin presentations with a little perspective. And so I like this slide. So if we think of it in terms of globally, we know we have about 10 to the 9th people living on the planet today. And within each person, there is a gut microbiota, and it is about 10 to the 14th cells within each person. So every person on the planet actually has more bacteria living in their body than there are people on the planet. And if we're talking about specifications in terms of the gut microbiota versus humans, the gut microbiota outnumbers us in number of species. They are, as many or more than the human cells in your body, and their genetic capacity is so much more than the humans. So you have this enormous community living in you, and the question is, what are they and what are they doing? So the gut microbiota is a complex community of microorganisms that reside throughout the gastrointestinal tract. It does contain an array of microbes, so bacteria, viruses, fungi. However, I will say here that the bacterial community is the most well-defined and is often what people are talking about when they say gut microbiota. And that is what I'll be talking to you about today. I'll be talking to you about the bacterial population, although there are other types of microbes that live in the gut microbiota. Although this community resides throughout the gastrointestinal tract, we know that maximal density is reached in the colon. And the colon community is the most commonly studied, although there are researchers now looking into the small intestine community, but the colon community is usually more studied because it is more dense. So within the colon, the dense community is functionally active. We know that it's able to take these undigested materials that make it from the small intestine and break down these undigested materials and produce metabolites. The most common are short-chain fatty acids, acetic, propanoic, and butyric acids. And importantly, the metabolites produced by the gut microbiota, they can be used by the cells, bacterial cells surrounding or the mammalian cells in the colon, but these metabolites can also enter the bloodstream. And so by entering the bloodstream, these metabolites are able to affect organs distant from the colon. So we know that the gut microbiota doesn't just affect the local area, but it's also affecting other organs far away, such as the brain. You know, there is a gut-brain axis. So this really shows us how the gut microbiota can be linked to health. So it is considered that if you can maintain or keep the gut microbiota intact, we can keep health, like a healthy outcome, where disruption or dysbiosis of the gut microbiota is often linked to disease. I will say here that whether or not the changes in the microbiota cause the disease or the disease is causing the changes, that is something that is not as defined, but we do know that we have observed changes in this community that parallel different types of diseases. So because we know that the gut microbiota can be affected by diet, and we know that the gut
Jenny, I'm sorry, but we lost your audio.
prebiotic, oh no,
You're back on.
okay. I'm sorry, I think my internet connection is unstable. And I apologize for that. So hopefully you heard a lot about the background here. But basically, where I'm going with this and what I'm saying is that, you know, research now is really trying to look at ways that we can intervene and we can help the gut microbiota either maintain health or potentially repair dysbiosis or prevent dysbiosis from occurring. And in that way, we can ameliorate these disease symptoms. So there's a lot of research looking at this right now because it's very easy to change your diet. So if you know that a prebiotic or a probiotic might help prevent a disease, then it's very easy to just, you know, add that to your diet. So while this is becoming more and more important to study, we have to find new methods to actually study it. And you might think, you know, I just said that you have 10 to the 9 people on the planet, you know, and each of them have a microbiota. It should be easy. And it's true, human subjects are easy to find, but they can be challenging to study in terms of the gut microbiota. And mostly this is because human subjects are really difficult to look inside. Your direct access is only going to happen with invasive methods, and most healthy people, especially, are not interested in being cut open to have samples harvested. And so for the most part, when we're looking at human subjects, we're looking at things coming in versus what's coming out. And this can provide a lot of really great information, but it doesn't tell us anything about what's going on inside here. So it can't answer questions regarding what and where and when things are happening within the gastrointestinal tract. Humans also can only be tested with safe products, which I mean does make sense. Like you can't just give a human subject something that could be toxic or lethal, which does mean that you have to have early stage development done on a product. So you can't just test an undefined compound. You have to have some kind of a safety profile, which does mean that you need to do some preliminary testing before you would actually be able to do a human study. You know, we do have lab animals that we can use to study the gut microbiota, and they can also provide some really great information. But with animals, you also have ethical issues. You know, again, with the whole safety profile, you have to have an idea of what it is you're giving a lab animal. And anatomical differences between humans and animals is a big problem with the gut microbiota because they tend to have communities that are different than ours. And we've actually have a paper that we published a few years ago where we looked at the gut microbiota of C. difficile because we had thought potentially to use this capuchin model to study the gut microbiota. And what we found was, although the capuchin is similar to humans in a lot of ways, the gut microbiota was very different than humans. And so it did show that, you know, if we're going to use an animal model, insight into some of these processes are still going to be difficult just because of the anatomical differences. So, you know, in vivo studies can really offer a lot of great information, but the one thing you're never going to be able to do with an in vivo model is remove mammalian components. And why is that important? So it's important because we know that within the gut, the microbes and the human cells or mammalian cells have this multifarious interaction occurring where you have the gut microbiota producing metabolite that can be used by a cell to produce another metabolite that can be used by the gut microbiota. So you have a ping pong effect occurring between your microbes and your mammalian cells. And it makes it almost impossible to know what cell is doing what. So when you're seeing an outcome, is it from a mammalian cell or a microbial cell or some sort of interaction? And this is really key. And because of this, it does mean that we need to develop some in vitro methods to look not only at what's going in this box here, the when and where, but also things that we can do so we can know what are the microbes themselves doing. So when it comes to in vitro methods, there are multiple methods that can be used, and they do range in complexity, and each of them have advantages and disadvantages. And this is what I'm going to talk to you about today. I will start by saying that the most elementary method to study the gut microbiota would be a single strain analysis. And in this type of a study, we would see a specific bacterial strain. You can either isolate it or you can order it from ATCC. Usually you would select a type strain, but you're taking one specific strain that is known to be part of the gut microbiota community, and you're going to culture it and grow it and test it on its own. And so you can see here is just a little depiction where you have individual cultures that you would grow and test. And this type of method is really good at looking at selection. So if you wanted to know whether or not a particular compound affects a particular bacteria, this is how you would do it. It's also a good way to look at gene expression because it is much easier to study the transcripts of a single culture than it is to look at a community. And we have done this a few times. So in the past, we've published a few different papers where we've looked at different polyphenols, so quercetin, naringenin, epigenin here on the left. And we've tested how these are affecting specific strains of gut microbes. And we've looked at it in terms of growth and transcriptomics. And really the results did show that, the effect and the response of each of these bacteria were dependent on the polyphenol and the bacteria itself. So they all responded differently to the polyphenols. And I'm not going to go into details on these results today, because I really want to talk to you about community studies. But I did want to point this out that if you're doing microbial studies, this is the most elementary method, and you can get some very good information, especially on specific strain responses. But you always have to remember that if you're looking at a single strain on its own, in reality, that strain is not going to be on its own in the gut. It's going to be part of the community. So today I'm focusing on talking to you about methods to study the gut microbiota as a community and not a single strain. And I'm going to go through today and I'm going to give you an example of 3 specific methods that can be used. These range in complexity, so going from simple to complex. And I'm going to show you some results that we have studying the gut microbiota using each of these methods. So the first method that I'm going to talk to you about is what you call a batch culture method, which is, your number 1 here. So it's the most simple method that you would use to study this community. So a batch culture method would be a single culture. So if you're studying the gut microbiota and you're going to use a batch culture or ex vivo method, basically what you're doing is you're taking a flask or a tube, some kind of a container. You can see these pictures here on the right. You have test tubes and you have a flask. These are very common things you find in a lab. And you're going to fill it with media and you're going to inoculate it with fecal material. So it's a very simple way to grow the gut microbiota. In this type of a method, you have limited environmental control. So you would be able to culture this under anaerobic conditions. So you could make oxygen-free media. You can pH the media. You can perform the work in an anaerobic chamber, and you can regulate temperature and agitation. But this is really the only type of environmental controls you have with this method. If you think of it again, it's a single flask that you're going to grow bacteria in. So because of the limited environmental controls, this type of experiment is short. It's usually 24 to 48 hours. I've seen some where they've extended them to 72 hours, but I think that that is really a bit long, but could be done. But either way, you're looking at an experiment that's only, 1 to 3 days. And it's because you have a buildup of metabolites and gases, you don't have any nutritional supply. So the bacteria in there are going to, when you inoculate them, they're going to go through exponential growth. They're going to produce metabolites. And then without any new nutrients being added to the system, eventually they're going to go into stationary phase and death phase. And so really this is the lack of environmental controls is going to limit the time that you can culture it. This type of experiment is often also called ex vivo. Whether or not you want to consider it ex vivo, I could leave that up to you, but it's called ex vivo in the sense that you're taking a sample harvested from a human and you're going to grow it to where it has limited compositional changes or limited modifications. So that's why some people will say this is ex vivo. So some advantages. So some advantages of doing like a batch culture experiment for the gut microbiota will be the short time. So, being able to get your results within 3 days is great. It's really good for screening. You can screen a number of cultures and have less product used. And we can capture inter-individual variability person to person. Your disadvantages here are not having a mature community. So as I mentioned, a few seconds ago, when you inoculate the feces, it's going to undergo exponential growth. So inevitably, you're going to have the faster growing bacteria, they're going to shoot up, and your slower growers are going to lag behind. This does mean that you're going to need to have a control when you're growing this so that you can look at what's happening over time and compare it to control. Obviously, we don't have any environmental controls. We can also only do linear kinetics here, so you can only test the effect of a compound and not its removal. So if you wanted to look at the before and after adding a food group, doing this type of method isn't going to work. And then I did put down here that, fresh feces are good for best practice in this case. And I know that doesn't really sound like a disadvantage, but it's not always easy to get as many fecal samples as you want at the right time when you want it. And so I did put this down as a disadvantage. Okay, so I'm going to actually show you a little bit more background on what I mean about individual variability and why it's important. And then I'm going to show you some results that we have using this method to look at lactose in the gut microbiota. So, if you have 7 billion people living on the planet and each person has a community, that is like I said before. However, each community to each person is completely unique. And this is because your gut microbiota develops in concordance with the host over a lifetime. And the type of community in each person develops based on their genetic background and things like diet, medication, supplements, like what are you putting in? And this does mean that you have a different community within each person that is important. It's definitely important to the person because it means that your community is specifically developed for you, and that's great. But when it comes to doing experiments and drawing conclusions, it does make it a little more difficult because not everybody has the same starting material. So understanding how a community is going to react to something when you have 26 different starting communities, it is not always easy to elucidate. In vivo methods can be applied, but again, you're looking at in versus out and not necessarily what's going on with the microbes. So your in vitro methods here do allow for more flexibility, like I had said in the previous slides. And in this case here, you would take your fecal samples harvested from each person. And if you're doing a batch culture method, you would just inoculate a tube and you could have 26 different communities here that you're studying. Each of them are going to be different from each other. And you're going to be able to take all of this data together and combine it. And you're going to be able to do an in-depth analysis looking at what the microbes are doing and potentially, not only what are they doing, but how could that potentially affect human health? And we're going to be able to identify donor-independent responses. So using all of these cultures, we're going to be able to screen them and say, okay, what is the response that occurs most of the time? So what responses occur no matter what your starting material is. And it's really important when you're developing something like a prebiotic or a probiotic, you want to know, is it going to work on those people and how is it working? So I'm going to show you an example of one of our studies that we just ran, looking at lactose on the gut microbiota using a batch culture method. So lactose is, I think everybody knows what lactose is or has heard of lactose. It's a compound that's only found in milk, which is a very important source of nutrition, and milk is considered part of a healthy diet for not just babies and children, but also for adult humans. Chemically speaking, lactose is a disaccharide of the lactose and glucose, shown up here. And if you are eating dairy, you are consuming lactose as part of your diet. You can see lactose here, it's entering the gastrointestinal tract. Then normally what occurs in the small intestine is lactose is hydrolyzed by lactase into galactose and glucose. And then these compounds produced are usually reabsorbed by the mammalian cells because they can use this as food. So this is what happens most of the time when you are consuming lactose. However, lactose does interact with the gut microbiota. First of all, you do have gut microbes that live here in the small intestine. It is not as dense as the colon, but they are important, and research has found really the small intestine community is important in digestion and absorption of food. But it's not as well studied, although you're going to find more and more that people are looking at small intestine microbes, that it is actually something that we are working on as well. But besides the fact that you have gut microbes living in your small intestine, studies have shown that actually not all lactose is broken down in the small intestine. So depending on how much you're eating, a percentage of it will enter into your colon. And, this is kind of important because bacteria also can break down, lactose. So when you have your lactose entering into the colon, it's meeting up with the gut microbiota, and the gut microbiota can also perform this reaction and produce metabolic byproducts. Now, most of the time, people will say, if you're lactose non-persistent and you eat lactose, you get uncomfortable feelings. And a lot of this is due to this interaction that occurs with the gut microbes and can have uncomfortable side effects. And it has increased a demand for lactose-free products because there are a lot of people who are lactase non-persisters. However, recently there have been a lot of studies that show that actually lactose may help maintain a healthy gut microbiota. And so potentially lactose could be considered as a prebiotic because it's able to help the gut microbes and swing it towards a healthy state. So the study I'm going to show you today is one where we took 18 fecal samples from individual adult donors in between the ages of 25 and 70. And if we look here at just the profile at the gut microbiota community of these donors, you can see that they are each different from each other. So they were completely unique. We did two groups. One had no treatment with lactose and one had lactose treatment. And so we're comparing each of these donors' feces to a lactose to a control to see what is happening. We cultured it over 24 hours and we took samples looking at community structure and functional output. And combining them together, we were able to identify some donor-independent responses to lactose. So I'm going to start by showing you a few examples of the community structure. So we took the samples and we ran 16S sequencing, and we looked at just the phylum levels of each donor with and without lactose treatment. So you can see at each of these time points here, you have a positive and a negative. So negative is no lactose, positive is lactose. And really what we saw from here was that there was a change occurring, and most of it was apparent within phylum Bacteroidetes. But really what we saw was that the magnitude of change was donor dependent. And so this is what I was talking about when I was saying that it was, we have variations occurring donor to donor. So when you're looking at a response of the community to a product, part of it is going to depend on the starting community. So there were some changes that are occurring that were donor independent. And so this kind of made us very interested to look a little more and see which specific taxa were actually affected by lactose. And can we identify a donor independent response or a response that occurred in most of the donors? So if we're looking at the results of our community analysis, what we see here is that at a phylum level, we had a significant decrease in phylum Bacteroidetes when we added lactose to the community. And this is important because your Bacteroidetes group, these are the ones that are usually degrading the carbohydrates. And so we did see a decrease in these. When we looked at phylum Firmicutes, we saw that there were actually quite a bit of changes that were occurring. We saw a decrease in a number of taxa, but the interesting finding here was that we actually saw an increase in a number of taxa that were lactose consumers, so things that would consume the lactose, and a corresponding increase in taxa that would consume lactate. So you have your lactose consumers that would produce lactate, and then you have an increase in taxa that would consume the lactate. And so these are considered LABs. Most people have heard of those. A lot of these are considered as a probiotic. And so we see a general increase, and this was a donor independent effect. So this effect was statistically significant, and it was not dependent on the starting material. We ran qPCR looking for bifidobacterium, which is another taxa that is considered healthy for the gut microbiota. And we saw a significant increase in bifidobacterium as well in response to lactose. So we're giving lactose and we're seeing a pretty good response to the community in a donor independent manner. When we looked at function, we looked at levels of short chain fatty acids or SCFAs. And like I said, at the beginning of the talk, these are produced as metabolic byproducts from the gut microbiota. And we did see that the total levels increased due to lactose treatment, which I mean, I feel like that's pretty expected. You give some source of nutrition, you're going to see an increase in the metabolites. But the interesting part was that we saw an increase in acetate and propionate, which did correspond to our increase in velinolaceae, which are known to produce these two SCFAs. We didn't see any change in butyrate. And so what we're seeing here is that the lactose is swinging an increase in propionate and acetate, but not butyrate. We saw a decrease in branched chain fatty acids or branched chain short fatty acids. These are usually the end product of a protein fermentation or amino acid fermentation. And so we're seeing a decrease here, and that makes sense because we're not adding amino acids or protein. Lactose is a carbohydrate. And we also saw a very large increase in lactate. And this corresponds to our increase in lactate producers. And so it makes sense. We had an increase in taxa that can produce lactate, and we see an increase in lactate itself. So the last thing we looked at for the study were some environmental changes. So we looked at gas production and culture pH as we were growing these. And as you would expect, we did see an increase in gas production as we were growing the cultures. And when we added lactose, we also had some increase in gas. And we also saw a change in pH. So for our control, our no treatment, we can see the pH kind of is maintained across the board. But when we add lactose, we did see a decrease in pH. This decrease in pH was significant, which is most likely caused from the increase in the acidic byproducts. And I'm just pointing this out because, you know, when we're doing a batch culture method, like I said, you're not controlling the environmental parameters, which means that when you add something that produces an acidic byproduct, you have a decrease in pH. And this can skew the community. So, you know, the community structure, when you look at the effect of pH, we know that if you have a decreased pH, you will decrease Bacteroidetes because these are less acid tolerant. We also know that LAB strains are more tolerant to low pH. So when you're lowering the pH of the community during fermentation here, you're kind of skewing your results to a decrease in Bacteroidetes and an increase in your acid tolerant taxa. And we also know that Lowering your pH will end up in decreasing short chain fatty acid production. And specifically here, we did see, we actually saw an increase in acetate and propionate. So that's probably not due to the pH. So taken together, our batch culture ex vivo experiment looking at lactose in the gut microbiota actually came out with a really interesting story where when we added lactose to the community, we saw this donor independent response where we have an increase in taxa that are consuming lactose and producing lactate. And these taxa are producing lactate, and the lactate is being consumed by lactate consumers. And these lactate consumers are producing acetate and propionate. And these increases in our short chain fatty acids actually drive a decrease in pH. And that decrease in pH can also affect the community structure. So it's basically the circle of effect. But I will point out that your LAB, your increase in your healthy bacteria, are considered as health positive, and an increase in short chain fatty acid is also considered as a health positive outcome. And so our conclusion from this study was that lactose is able to promote an abundance of healthy bacteria and increase the levels of beneficial metabolites. And this manuscript is actually currently under review. All right, so I'm going to move on. And I wanted to talk to you a little bit about studying the gut microbiota, not just using a batch culture, but also using what we call a single vessel system, where you're using one vessel to grow a community. So a single vessel or bioreactor system will typically involve the application of some kind of a computer-controlled equipment. So you can see here on the right, this is an example of one of the systems we have in our lab, which is the Eppendorf Bioflow 320. But of course, there are many types of bioreactors out there. So not just Eppendorf. So usually you will have something that is computer controlled in order to control the environmental parameters. So in this situation, you would have your reactor and you'd fill it with media, inoculate it with fecal material. The computer is normally set up to maintain the community over time, and it's automated. And it can control things like temperature, pH, agitation, gas. And so if you see here, this is just a cartoon depiction of a bioreactor where in here you'd have your gut microbiota growing. And you can see that we have now an inflow of media. So you're going to actually feed the system with media and take out waste. And so now you're looking at every 8 hours, you're cycling. So you're feeding the media, feeding the culture and removing waste every 8 hours. You could potentially do continuous feeding where you're continuously feeding and continuously removing. But when we set these up, we typically like to use 8-hour cycles because that is what a human would do. You know, you would normally eat every few hours, 3 times a day. So because you have these control of this environmental parameter, we are able to develop what we call a mature community. Meaning that this gut microbiota here, when you inoculate the fecal material, it's going to undergo exponential growth. And then over time, the community is going to, they're going to fight, right? So there's competition that's going to occur. And then eventually they're going to reach a steady state where the microbes themselves are not going up and down, but they're maintaining that community. And a mature community will remain stable unless it's disturbed. So once it reaches stability, as long as you're feeding it and removing waste using your daily cycles, you're going to have this community stay the same. Now, it does take a longer time. It takes about 14 days for a mature community to develop, although you could decrease this if you change your retention time. But mostly we consider 14 days for a mature community. So if you're using this type of a system, some advantages would be that there's a simple design. So you're just using a bioreactor and you're going to, add stuff to it and grow it up. We are using a mature community. We do have control of parameters and we can test response and recovery. So you can add something and look at what's happening before and after. And because of these advantages, we've actually have a number of papers that we've studied using this type of a method, looking at how different food components affect the gut microbiota. Disadvantages would be the time, because now we're talking about weeks and not just a few days. And it's a larger volume because now you're talking about a bioreactor. And you're also limiting the number of donors that you can test. So if you are looking at these larger systems, you're not going to screen 20 different individuals. You're looking at maybe 2 or 3 at a time. So I'm going to show you an example of some of our work where we used a single vessel system to study the effect of pectin. So most people have heard of pectin. It's a plant cell wall polysaccharide, and it's distributed throughout the plant cell. And we can see here it is found in ubiquitously in fruits and vegetables. And the type of pectin that is in each of these is categorized based on 2 parameters: molecular weight and degree of esterification. And a degree of esterification has to do with the molar ratio of methyl esters that's present. And a low DE would be less than 50%, and a high DE is greater than 50%. And so when we're looking at pectins, we know that, again, pectin being part of a cell wall, it's found in all your plants. And the type of pectin in each of these differs based on its molecular weight and DE. Importantly to us, because we are gut microbiologists, pectin is consumed as part of a diet, or at least part of a diet that contains fruits and vegetables, right? So if you're eating fruits and vegetables, you're going to get pectin. And the good thing about pectin, if you're looking at it in terms of a prebiotic, is that it's not affected by enzymes and saliva or gastric juices. And so it's not actually degraded or used by mammalian cells, but it does make it through the colon where it contacts the gut microbiota. Now, the gut microbiota can ferment pectin and produce short-chain fatty acids. And so this is why pectin is considered as a prebiotic, because your bacteria in your gut are able to use it to produce healthy metabolites. And, pectin itself is directly affecting the gut microbiota. So I'm going to show you some results from a study where we looked at how pectin structure actually affects its prebiotic potential. So in this study, you can see here, we used 2 donors, which is much different than that first study where we used 18. So we had 4 bioreactors that we used, and 2 of them were for donor 1 and 2 of them were for donor 2. And so we used these bioreactors and we tested 2 different types of pectin. We tested lemon pectin. One of them was HMW-LDE, or high molecular weight, low degree of esterification. And the other pectin was a low molecular weight with a high degree of esterification. So we were testing 2 different types of lemon pectin on 2 different donors. And so what we did is we cultured the gut microbiota to develop a mature community. After that, which was about 14 days, we started to add our lemon pectin. So we had treatment. We added 1% lemon pectin for 3 days. And then we stopped treatment and we did 9 days post-treatment. So we were able to look at the effect of pectin before. So like before what was happening in the community, we add lemon pectin, we saw what changes occurred. And then we were able to see after we stopped adding the pectin, what changes occurred. And we looked at this in terms of community structure and short-chain fatty acid production. And so I'm just going to show you some basic results from this study. So we found that your low molecular weight, high density pectin actually induced a donor-dependent response. So what this means is that the donors responded differently to this pectin. So donor 1, we had a decrease in Veillonella and Prevotella and an increase in Eubacteriaceae. Whereas donor 2, we saw decrease in Rickenella and an increase in Raminococcaceae and Fusobacterium. So in these figures, your donor 1 is shown in gray and donor 2 is shown in peach. And so what we can see is that the communities were responding significantly when we added the low molecular weight, high degree of esterification pectin, but it really depended on the starting community. So the different donors responded differently. When we looked at the high molecular weight, low degree of esterification pectin, we did see a donor-independent effect, meaning we saw that Lactose increased in both donors. Although the specific type of Lactose did differ between them. However, this does show that this type of pectin, or this lemon pectin, was producing a response that was more donor independent. So whereas donor 1, we only saw a change in Lactose spraceae, donor 2 actually had changes in multiple taxa. So donor 2 was responding a little bit differently than donor 1, which is expected. And some changes did occur after treatment and during recovery, which is again another benefit of this type of system, is we can look at the recovery that happens as well. When we looked at short-chain fatty acids, what we saw was that both pectins were increasing the production of short-chain fatty acids, and the increase actually persisted, especially for our donor 2. So even when we took the treatment away, we're still seeing this increase. We looked a little bit more at which short-chain fatty acids were the ones contributing to this increase. And we found that although short-chain fatty acids were increased in response to both types of lemon pectin, the specific types of short-chain fatty acids that were responding differed. So for our low molecular weight, high density, or high degree of esterification pectin, we saw an increase in butanoic and pentanoic acids over here, whereas for the high molecular weight, low degree of esterification pectin, we saw an increase in acetic and propanoic acids. So they're actually responding by producing these different types of short-chain fatty acids. And so this manuscript is actually also currently under review. And our conclusions here were that the molecular weight and degree of esterification of the lemon pectin did elicit different responses. So if we look at the 2 types of lemon pectin on the gut microbiota, what we saw was that the structure was affected differently depending on the lemon pectin. So whereas our low molecular weight, high degree of esterification had a variable response, our high molecular weight, low degree of esterification had an increase in Lactose spraceae. When we looked at this in terms of function, we saw that both lemon pectins increased function by increasing total short-chain fatty acids, but they were doing it through alternate pathways. And so the type of pectin was kind of tailoring the response of the gut microbiota community. Okay, so the last thing, I have about 15 minutes left. I'll try to move through it quickly. I did want to go over multiple vessel systems. This is our 3rd type of in vitro method that we use to study the gut microbiota, where we're using multiple vessels to simulate the entire gastrointestinal tract. So like, why do we need to do that? So like I said in the beginning, the humans are very difficult to look inside of. And when we use fecal samples or even those single culture experiments, like I just showed you, you're only looking at input versus output. And really, you don't have any internal resolution that occurs telling you where in the colon something might be happening. And in this case, when we use a single vessel, you're considering the colon as one, right? It's a whole. It's just this one big box. It's a single unit. And this is great for screening, and it does give us some good information. However, in reality, we do know that the colon is not a single unit, that there are differences that occur within your different colon regions. So you have your ascending, transverse, and descending regions. And there are different communities that develop because we have different environmental conditions in each of these regions. So we see first that nutrients are flowing from the ascending to the transverse to the descending. So we have a flow of nutrients that occurs within the colon, and the nutrients that enter are metabolized in a cascade-like manner. So your ascending colon community is going to get these nutrients first, and it's going to produce metabolites that can be used locally, but also the metabolites produced upstream can be used downstream as a substrate. And so really what we see in the colon in a human is that there's a gradient that develops where we have different microbes and an array of metabolites. There were two papers written looking at sudden death victims and looked at their colons and what was happening. And they looked at proximal versus distal. They didn't break it up into the 3 regions. But even looking at just proximal versus distal, what they found, I see you, thank you. I know I have 15 minutes, yes. Okay. So what we see is that in the ascending colon, we have more fermentable substrates that are present and more short chain fatty acids produced. And by the time all of this stuff reaches your distal colon, you end up having more protein breakdown, amino acid fermentation, more branched chain fermentation, branched chain, short chain fatty acids that are being produced. We also know that within the colon, we have two types of distinct communities that occur. We have a community that is within the colon, or what you call a luminal community, and then you have a community that grows within the mucosal layer of the colon. And these two different communities are really tailored for different environments. Specifically, your mucosal community is going to be able to actually consume mucin. So it's going to be able to consume the mucin and usually survive a lower amount of oxygen, considering that it is believed that there's an oxygen gradient due to the blood flow within the human cells. So when you have a multiple vessel system, it usually tries to take all of this into consideration. And so today I'm going to talk to you a little bit about some of these multiple vessel systems. And I'm going to try to move through pretty quickly so I have time for questions and answers. But in this type of a multiple vessel system, we see that each of these vessels are hooked up together. So you're simulating your entire gastrointestinal tract. And this was that same figure for your single vessel system. But in a multiple vessel system, you have a bioreactor for each region instead of just an individual bioreactor. We're also still maintaining our daily cycles. And with a multiple vessel system, we can actually add in a mucosal community by adding in these little mucosal carriers that can grow the gut microbes themselves. So you also now will have a luminal and a mucosal community. And this does take longer because this type of system is more complicated. These experiments are usually 4 to 8 weeks in length, not your 1 to 2 days or 2 to 4 weeks. So, some advantages here is that you can look at different community structures. So you can look at luminal and mucosal communities. We can look at where a reaction is happening. And using this type of method, we have a number of papers that we've published. Most of our papers up until now have been looking at the system itself and seeing how the different communities in the different regions are more complex. Are we increasing in complexity as we go, or where are the differences occurring? And so obviously our disadvantages would be that it's complicated and long to run. And so I'm going to show you just a little bit more information on what the multiple vessel system is that we use, and some data on the different communities that develop. So we use the SHINE model, Simulator of Human Intestinal Microbial Ecology. However, I will say there are multiple types of models to use. This is just one. In this system here, we see that we're breaking up the gastrointestinal tract into different regions. So a bioreactor representing each of the regions. We have fluid moving sequentially from one reactor to the next, mimicking that flow of nutrients. We can add in food and digestive enzymes, 3 times a day, every 8 hours. And again, we're going to be able to simulate that mucosal community by adding in our mucin agar here. And this is just a picture of it. We maintain different pHs within each region to mimic what we know about pH of the gastrointestinal tract. And 2 of these together is called a twin SHINE. So you have 2 complete systems where you run them together. And this is a picture of the twin SHINE. So just a general picture of it in our lab. This is a picture of the mucosal community growing on the mucin agar. And this is a picture of the setup with the bioreactors. So my last few slides today are just showing where we've taken this multiple vessel system and inoculated it with a donor fecal sample. And we looked at which types of communities are growing in the vessels and the luminal and mucosal portions. And we used the same donor, and we actually inoculated a single vessel system. And so we compared the 2 types of systems as well. All right. So I know that I am running out of time. And so I just want to point out that when we have our luminal communities, we did see an increase between the ascending to transverse to descending. So when we're looking at the 3 colon regions, we see an increase in diversity as we're going through the system. And we also see this in the mucosal community as well. When we look at a single vessel system, the community is similar to your TC, which is kind of that middle community, which makes sense. If we're looking at a PCOA, looking at unweighted unifrac distances, so measuring communities based on presence or absence of taxa, what we found here is that your AC community was very separate from your TC and DC communities, and your TC and DC communities are very close to your inoculum. Your single vessel communities kind of cluster together, but far away, actually, pretty distant from the inoculum. And so we do see here that there's a big difference between your single vessel versus your multiple vessel system right away in terms of community structure. So what was driving this type of divergence? And I'm just going to point out that between the multiple vessel and the single system, really the big difference here is Bacteroidia. And so in your single vessel system, we had a decrease in Bacteroidia. In the multiple vessel system, we actually see that Bacteroidia are maintained pretty well. We also saw an increase in Proteobacteria when we're looking at the single vessel system, which we did not see in our multiple vessel system. For both of these types of systems, we did have an enrichment of Clostridia in the mucosal samples, which was really great because it's showing us that we have different communities developed in the mucosal versus the luminal communities. And the take home really for the community development is that nutrient availability was key, where our multiple vessel system is producing unique communities between the mucosal and the luminal phase. When we look at the production of short chain fatty acids, we'll saw that your single community, so that single vessel, actually had a lot less short chain fatty acids produced compared to our multiple vessel system, which had a lot more produced. And we did see a step increase in between the three regions where we had what we think is an accumulatory effect. So we're looking and we see the DC has the most, the descending colon had the most short chain fatty acids, but were they there because the descending colon was producing them, or are they just accumulating as it went? And really, when we looked at the presence of glycoside hydrolase genes, we saw the opposite, which indicated that the AC community actually has the highest potential to produce short chain fatty acids. And so what was happening in the system is we are having an accumulation occur, which is the drawback of this type of system. So we have short chain fatty acids produced in the ascending colon, and they are not being absorbed in the transverse or descending region. So the last thing we did was
Dr. Firrman,
yes.
we were hoping to have time for a question and answer. Would you be able to wrap up?
Yeah, I can definitely wrap up. This is actually published, so you can read this if you're interested. But when we looked at metabolites, we did see that we had distinct regional community profiles that developed, and we saw different types of metabolites produced in the different colon regions. And so we know that the types of metabolites are changing as we go. And we also know that if we do an analysis looking at structure versus function, that they correlate very closely, where we're seeing that structure and function are very similar to each other in this system. So we went over 3 different methods to study the gut microbiota, although I know there's much more I could talk about multiple vessel systems. And I'm just going to wrap up by saying, you know, research is still showing how we can study the gut microbiota, you know, to find ways to increase human health through dietary intervention. And this is my acknowledgement slide. So this is everyone who's worked on this project with us up until now. And I can take questions. I don't know, probably you guys have questions on the Scheim system. That's what I would guess. Do you want me to
Thank you so much. That was an excellent presentation. You're done, right?
Yeah, do you want me to stop sharing?
We can ask our people behind the scenes to work on that. So excellent presentation and discussion across these different methods. I'm going to move right into the question and answer. So Alice Lichtenstein asks, In the batch culture ex vivo lactose studies, you observed a donor-independent change in branched chain fatty acids. Was there a change in fatty acids with trans double bonds? We've lost your audio again, so we haven't heard your answer.
Oh.
Can you try again? Yep, you're back.
Okay, sorry. Okay, I would not be able to answer that without actually looking at the data. And my light has turned off. So I can't answer that. I would have to take a look. Unfortunately.
Jose Ordovez asks, what do you think about gut on a chip, where you can examine more complex interaction between microbiome, enterocytes, hepatocytes, for example?
Yeah, no, gut on the chip is really great. And I think that I've seen a lot of talks on that, and I think it's kind of where we're going, because we know that not just the microbes, we want to know how they affect the human cells. Like that is really key to its effect on human health. And so in those types of those gut on chips, we can look at that. From what my understanding, really, that one of the hardest parts of doing gut on chip is limiting overgrowth of the bacteria. And so in those types of models, when you're adding gut microbes into the system, they're able to kind of grow and overgrow on the human cells. And so that is, from what I understand, is one of the drawbacks. But besides that, no, I think that they're really great. And I've seen a couple talks on it. And it does make me like, wow, I'm really interested to see where, this is going to go.
Yeah, it's an interesting method.
Yeah.
All right. I'll keep going. So Chris Wiley says, fascinating work, getting closer to in vivo modeling using bioreactors. In these studies, are you using an anoxic environment, or are you simulating oxygen pressure found in the relevant gut areas? If so, did you see, sorry, I'll just finish it. If so, did you see changes in species enrichment based on the oxygen concentration used?
So normally we run anaerobic. And so we do set it up to be 0% oxygen. In the Scheim, it's nitrogen flow that is, so in the Scheim, those bioreactors are like sealed. And in the Eppendorf, it's continuous nitrogen flow, like a pump to keep it anaerobic. I would say that, when it comes to adding oxygen, we've done one study where we've added a little bit of oxygen into the colon, and the structure didn't change as much as you would think. But we don't normally do that for the colon community. For small intestine, I think it'd be a little more important to add more oxygen in, but not for the colon.
Okay. The next question comes from Shangdong Wang. Could the lactose treatment as prebiotic be problematic among people with lactose intolerance? Is it possible that other prebiotic compounds which modulate gut microbiome could improve lactose tolerance in people?
Yeah, so I mean, definitely if you're a non-persister, then if you are eating lactose and you could have side effects that would be uncomfortable. I think, we're in Dairy and Functional Foods Research Unit, and so we do try to look at milk and some dairy. And for me, I feel like because there are lactase non-persisters, there's a drive for more lactose-free products. And so people consume lactose-free products, and it actually is sweeter when you have if you have like a lactate where they break up the lactose before you drink it, it tastes sweeter. And so people do actually like the taste of it better. But interestingly, when they try to look at human genetics and compare it to the gut microbiota and see how your human genetics play a part in the type of community that develops. One of the things that they're constantly finding is that there's a big correlation between people who are lactase persisters or non-persisters, or people who are producing lactase, and bifidobacterium, which is a healthy gut microbe. And they find that people who are actually somewhat lactose intolerant who still consume a little bit of lactose in their diet have an increase in bifidobacterium, which is considered a healthy effect. And so I would say, you know, I wouldn't necessarily eat lactose if you're a non-persister, but if you're someone who can tolerate it, it's definitely something that is healthy for you if you're eating it, or at least it's having healthy effects.
Okay, we'll take one more question. This will be our last question from Chao Lai. So for the lactose experiment of 18 donors, have you examined the difference between LCT genotype, well known for genotype for lactose intolerance, of 18 donors for microbiome pattern?
We've not, and it would be really interesting to do that. But I think part of the question is getting that information from the donors after we've taken the samples, being able to go back and get that from them. So we've not, but it would be really interesting. And I know we're talking, so we did this with a collaborator who's able to harvest the 18 donors because we don't harvest samples at my site. But we've talked about trying to like go back and do this, and it is something that we would like to do and we're hoping to be able to do, but I don't know that we're going to be able to get that from those donors at this point.
All right, so this concludes our seminar. Thank you so much to our speaker today, and also thanks to the attendees for coming. So we'll wrap up. The next HNRCA Monday seminar is in 2 weeks, and we'll see you then.